All right, we're recording. So hi everyone, thank you for joining our TEM user meeting for AIF TEM users. Today we have the lovely Rohan Dhall, Dr. Rohan Dhall, beardless and ready to go. Rohan used to be a postdoc at NC State in my research group, and he was also a TEM lab manager for the analytical instrumentation facility. So we're excited to have him. He's now what's your title? Senior staff scientist extraordinaire?
Not at all.
Master?
Senior scientific engineering associate?
Ooh.
Whatever that means.
Yeah, at the National Center for Electron Microscopy over at Berkeley. And we're very excited to have him. So we're gonna have a nice little Q&A here. Yeah, I'm gonna keep everybody muted for now. If you do have a question that you want to ask out loud, go ahead. But if you want to just type in your questions in the chat throughout, definitely do that. This is pretty chill, just catching up with Rohan, and I don't know, we can learn stuff about what a staff scientist does at a national lab. Yeah, so Rohan, if you want to get started, just like tell us a bit about yourself and what you do at INSIM.
Cool. Okay, so thanks for having me here, and it's nice to see some old familiar faces. So well, let's see, let me start from grad school. So I'm actually an electrical engineer who got bored of electrical engineering as an undergrad and decided, I'm going to study physics. And so I did my PhD at USC in Los Angeles with a professor who was studying carbon nanotubes back when it was cool. And I still think carbon nanotubes are cool, but most of my PhD research work went into joule heating of carbon nanotubes. So we were making small electrical circuits in a clean room and doing nanofabrication, electrical characterization, that kind of stuff. And my advisor was good at doing optical spectroscopy. So we used to do a lot of Raman spectroscopy. So none of this ever had anything to do with TEM stuff. And I took a long time to graduate, but part of the reason why was somewhere in the middle of my fourth year, I met a very evil human being who said to me, hey, you want to see something cool? Come with me. I'll show you the TEM lab. And this literally happened. There's he's now works at Aerospace Corporation, but this guy basically took me to a TEM for the first time in my life, and they were tuning things up, and he had just gotten hired there. And I saw an atomic resolution image of cold nanoparticles. Yeah, I know, so boring and not particularly exciting. And I nearly pooped myself. And I was like, wow, this is cool. So I started coming up with all kinds of terrible ideas to try and get into the TEM lab. And I ended up wasting 2.5 years of my life on a research project, which I convinced my advisor and a professor at UCLA would be a great use of time and energy. It was a really bad idea from the beginning, but it also involved a lot of effort. So, you get the best of both worlds. But that gave me my first experience in electron microscopy. And honestly, I was still pretty much a novice microscopist at that time. But what happened was, by the time I graduated, I said, hey, I had gotten enough of a taste for the field that I thought this is something I should take a little more seriously if I want to have a career in this field. And so I ended up joining Jim LeBeau's group, who was at NC State at the time. And there were multiple reasons why I did that. One was because he offered me a job. That was nice. But the other reason, there were two key reasons. So I was very, very impressed by the facilities at AIF and at NC State when I first visited, which was 2016 or maybe late 2015. I was really impressed by the kinds of research that Jim was doing at the time. All of his students at the time and still are very, very impressive people who know what they're doing. So I thought, this is a great learning opportunity for me to, actually become a real microscopist, as opposed to just somebody who likes to use the TEM like a point and shoot camera. And so it turned out to be a great decision at the time, because I think Jim gave me a lot of freedom in doing what I wanted to do. And the second big reason why I was interested in coming to NC State was, so a lot of what I had done thus far was in situ TEM, because even when I was an optical spectroscopist, what I used to do was electrical measurements under an optical microscope. And I was interested in carrying that forward. And NC State has a big advantage in that it's co-located. It's next door to, I guess, Prototypes is next door to NC State. So I thought this would be a good way for me to learn a little more about that subfield of electron microscopy. And it turned out to be a great decision, because the folks at Prototypes are really helpful and always encouraging new, stupid ideas, and give you a lot of good feedback. So I think from that perspective, it turned out to be a really good decision. And then when did I quit? Not quit. 2018, I guess. So it's been like 2 years is approximately when Jim decided that he might be moving away from Raleigh. And so I sort of panicked and I started looking for jobs. And this job had literally just been posted online that very night when he told me that you should start looking for jobs. And I applied and I came in and interviewed, and I really liked the vibe of Berkeley Lab. And I started work here sometime in the middle of 2018. And the one other advantage for me was that as a PhD student, at some point we realized, the electron microscopy project that I had proposed was not so smart, but it could possibly be done on a special, not a special, but a certain kind of microscope, which was at Berkeley Lab at the time. And so I was able to visit as a user when I was still in grad school. And I had a feel for what life was like in Berkeley Lab, what the work ethos and work culture is like. And also, I just sort of knew what I was getting myself into. And those were big motivating factors for me to take this job, because my first experience with Berkeley Lab had been a very, very positive one, even though the research results, I had no research results to back up that entire effort. But I still walked away from it thinking, it's not because of anything that Berkeley Lab did to hinder my progress. They actually helped a lot. And so that's why I wanted to be part of the same kind of work culture if possible. And I'm quite enjoying it so far. So that's my brief, not so brief bio.
That's great. I appreciate that. So like now that you're on the other end, now that you are the person that's helping us that may not know exactly what we're proposing, do you have any advice for those of us that may be submitting proposals of like what we definitely want to look into, or what you look for in these project proposals to like get them accepted?
Sure. So I don't know if everybody's aware of the way the NSRCs work, but basically the Department of Energy funds these research projects, which are fairly, big, like multi-million-dollar projects, which are supposed to be open user facilities for anybody and everybody to come and do science if they want to. Typically, these are divided into, a lot of them are synchrotrons, and that's because partly political and because of who has traditionally been in charge of the Department of Energy over the last 30 or 40 years, and the people who've been in those positions have wanted to see more synchrotrons. But the other reason is a lot of DOE labs were started during the Manhattan Project, when nuclear science was a big focus of everybody's research. And that may not be the case so much anymore, but basically the first thing a lot of these labs ended up building was a big radiation source. So Berkeley Lab, for example, has something called the advanced, it has a big synchrotron. What is our synchrotron called? All the synchrotrons have weird names. So the advanced light source, that's right. I had a brain fart. So the advanced photon source, I think, is at Argonne. Stony Brook has its own accelerator. SLAC is the one at Stanford. And so there's a whole variety of these. But Berkeley Lab, the division that I work in is called the Molecular Foundry, which is predominantly involved in nanotechnology research. So we have several floors, and each floor has its own kind of competence. There's a floor for theory people, for example, so people doing DFT calculations. There's a biological nanostructures floor where there's all kinds of experts who are making small biomolecules and looking at what they do in self-assembly and stuff. There's an inorganic nanostructures floor, which works a lot on like nanoparticle synthesis or maybe lead halide nanocrystal synthesis, things like that. There's a nanofabrication floor, which is just doing clean room nanofabrication. And then there's one floor, which is called the imaging facility, which basically has a lot of really nice optical microscopes, although they also have the TEM that I worked on as a grad student. And then there's our floor, which is not a floor, but a whole separate building, which is the National Center for Electron Microscopy. And essentially, if you are a user and you are a prospective user and you're interested in doing some kind of research at Berkeley Lab, you need to firstly figure out what are the facilities that are available at Berkeley Lab and how those can meet your research needs. And then twice a year, we have proposal calls where you are asked to submit user proposals, which then go through an independent peer review, where we as scientists have no control over whether or not your proposal gets accepted. But every proposal gets scored on a rubric. That rubric is all available online. And then once you get your score, there's a cutoff determined by the facility directors, based typically on our just how many hours there are in a given week and how many staff members we have and our ability to support projects. And then if you meet those criteria and your proposal gets accepted, you can use all the facilities at NSEM or at the Molecular Foundry for free. It's completely free of charge, and you get access to the Berkeley Lab staff and their expertise, which is oftentimes very helpful. Now, the one thing I would say is it's always a good idea to get your proposal vetted by a member of the Berkeley Lab staff before you actually submit it. And there's 2 or 3 reasons for that. So firstly, we can point you to obvious issues with your proposal. Just a hypothetical example, let's say you want to work on our THEMIS image corrected TEM, and you write somewhere in your proposal that I want to do probe corrected stem eels on this microscope. Now that proposal is likely going to be rejected straight out of the bat because you'll get a 0 on the feasibility score because the THEMIS microscope does not have a probe corrector and doesn't have an eel spectrometer on it. But those are, if the scientific case is solid, that should not be a showstopper, because at NSEM we have so many microscopes that we can point you to the correct resource, right? So if I see a proposal like that, the first thing I'll say is, hey, you should change the word THEMIS to TEAM1 and everything else stays the same. And TEAM1 is just a different microscope that has those functionalities on it. And then you don't get rejected for it's a scientific reason, but in my opinion, that's always a sad reason to lose out on an opportunity. The other reason why I encourage you to reach out to staff members is, so as members of scientific staff at Berkeley Lab, our job is to help anybody who has an active proposal. So it doesn't matter if I think your research is the coolest thing in the world, or if I think, I don't particularly care for that research project. I mean, I'm still going to help you, and I'm going to give you the best advice that I can to help you get to the kinds of results that you want. But if you can find some overlap with the interests of existing staff members at Berkeley Lab, I don't think you have a massive advantage in terms of getting access to the microscopes, but I think it does help foster collaborations that can be very, very productive. So a lot of the members of NSEM staff collaborate with their users, where they're always on joint publications, or often on joint publications. And it just saves users the time and energy of sort of reinventing things that somebody at NSEM has already done and is an expert at. So just off the top of my head, like if you want to do tomography, and we have a guy called Peter Ursius on our staff who is like a tomography legend, and so why would you not make use of his expertise and knowledge set? So that's why I recommend that people reach out to staff members before the proposal submission. The deadlines for proposal cycles, I think there's one in around March, April, there's one in October-ish. These are all posted on the Molecular Foundry website. And I recommend not to wait till the last week before you submit a user proposal, because that's when the staff get flooded with requests to review proposals. Often that's like, the last week of each scientist would have something between 10 to 50 proposals at their desk, which are not very long. They're like 2 to 3 pages long, but it's still, that in addition to all the regular work that you're doing might mean that that you can't do justice to each of those proposals. So I recommend, even if you miss the deadline, go ahead, write your proposal immediately. There's no harm in submitting a proposal very early. And I think that way you get the best feedback.
Awesome, thanks.
Mm-hmm.
So I will pause for a moment and ask if there are any questions from the crowd, if anybody's dying to ask Rohan anything. Abby, or yeah, let's go Abby first.
Okay, hi Rohan. Okay, so I have kind of a weird question for you. What skills have you learned through electron microscopy that have been useful in unlikely places? And you can interpret that question how you will.
So these are not skills that I picked up in at NSEM necessarily, but there's a lot of technical skills that you sort of need to be a good electron microscopist. So I told you I used to be an optical microscopist and an optical spectroscopist long ago. When I first started doing TEM, I thought TEM people were spoiled. And this is because everybody with a big TEM has a giant service contract, and they paid $5 million for this microscope. So they pay $100,000, and every time something goes bad, some magic human being pops up and fixes everything for you. And it's like, wow, this is like, you're doing nothing, because 99% of my time in an optical spectroscopy lab was basically spent ripping apart the whole microscope and putting it back together, which happened once a week. So I think from the technical skills standpoint, now, that said, I now realize the value of that, because I don't think it's feasible to rip apart electron microscope once a week. But I think a lot of software skills are becoming more and more important for students and postdocs to have, just because the immense amount of data that people are gathering and then analyzing the data afterwards. But really the most important skill that I've learned, I think, is effectively dealing with people. because I feel like electron microscopy is often a little more collaborative than a lot of other branches of science. Like just the nature of research groups is like, oh, somebody in that research group comes and gives you this crystal. You make a sample, you look at it in the TEM. So there's always this back and forth between different research groups. If you're a grad student, for example, and this is a little hard to put into words, but I strongly believe that the personality or identity of the principal investigator in a group sort of permeates through the entire group. So I found that a lot of my lab mates were very similar in their temperament and such to my PhD advisor. And that makes it very easy to interface with your own lab mates because you're kind of like-minded. But sometimes when somebody from a different lab shows up, it makes it really hard to communicate and collaborate because you just can't understand why their priorities are so upside down. And I think doing more TEM work has given me a little more patience about that and helped me try and see things from a different person's perspective. And that's a useful skill in life, I think, in general.
Patience is definitely a very useful skill.
Yeah.
No, I can see what you're saying about the personality of the advisor carrying through. It's definitely, I can feel it for sure.
Yeah.
Abby, thanks for your question. Chris had a question, and then I'll go to Shadow. You raised your hand next.
Rohan, I was wondering if you could talk a little bit more about the kind of projects you started working on when you went to INSIM and how the projects have evolved. And I guess my main question is, how do you see TEM STEM techniques? How have they evolved in terms of what the user base is asking for, what kind of scientific questions they're trying to answer using the kind of hardware you guys have?
Mm-hmm. So those are both good questions. I think there's a lot that I can say. So personally for me, when I moved to NSEM, the biggest component of my portfolio was not one specific microscope, but it was all the in situ holders that we have at NSEM, which, you know, if you add up the value of each of those holders is probably 3 times more than one of our nicer TEMs. So this is a guesstimate because we just have a lot of them. And so I, being an electrical engineer, I think I was a little more, little better trained to take over that task of, you know, running all the in situ holders. And so that was the first thing that I started off doing. However, the boundaries at NSEM of what you are allowed to do and what you're not allowed to do are very, very blurred. So if I had a personal interest in learning tomography, for example, all I had to do was walk up to Peter and say, hey, would you spend 3 hours with me on the nicest, coolest microscope in the world for doing atomic resolution tomography? And he would probably do it because that's the kind of work culture at NSEM. But what happened with me was there was a new microscope that had sort of just been installed called the TEMIS, which is very similar to the NC State Titan. By the way, I have to say this. I think the NC State Titan is still to date my favorite microscope of all time. And it's just for a bunch of personal reasons and also because I think it's very, very well configured. So anyways, the TEMIS was the closest thing we had to that. And there's a member of staff who was already in charge of sort of maintaining that microscope. But she is probably the most overextended member of staff that we have. Her name is Karen Bustillo. She is an amazing person to work with. But you know, when I came in, I said, hey, if I'm here, I can help you on this microscope, for example. And so it ended up being that I was doing a lot of work on that microscope to begin with. And from a technical standpoint, it started off with, so that microscope has a SATA camera, which is a really nice, reasonably high speed bright field TEM detector. And so we started out doing some liquid cell imaging, which was where, a high-speed detector can be really useful, because in a liquid medium, your nanoparticle or whatever is always moving around. And those were the first sets of projects that I think I really started working on. Then I got good with all the indentation holders. And so the nano indentation holder is a really interesting thing. If you have never done nano indentation, it's a whole world in and of itself, but basically it's trying to study mechanical properties of materials under a TEM. So you can poke a nanoparticle, for example, and see how it deforms. And we have a couple of those kinds of holders. And so those projects were very interesting to me at the beginning. I did a few biasing experiments, which sort of did not go anywhere for a variety of reasons. But these are things we are still working on. But then the other thing about that microscope is it also has a Lorentz lens on it, and that was somewhat underutilized. In fact, when I went there, I don't think we had a single user who was doing Lorentz microscopy. And so I said, hey, we have this thing, I should learn how to use it. So I started emailing people at people who knew how to do Lorentz microscopy across the world. A lot of them were in Thermo Fisher. And then I figured out how most of it works, and we were able to do a couple of really cool projects. So the big example I give people is there was this study still under review on a material called FGT, which is iron germanium tellurium or telluride. And when it's cooled down within a certain temperature range, it shows, so it's a magnetic material which shows magnetic domains at even at room temperature. I think the Curie temperature is just a little higher than room temperature, like 350 or so Kelvin. But then when it's cooled down, it starts showing skyrmions within a certain temperature range. And so that was kind of the first really interesting project that I got involved in, where we used a liquid cell, a cryo holder to cool these samples down to liquid nitrogen temperatures and observe what happens to the magnetic domain structure using Lorentz microscopy. And then we were also obviously able to tune the Lorentz lens or the objective lens current to increase the magnetization and see if we can drive away this skyrmion state, which we could. So that was kind of the first super exciting thing that I think I did. And but that's not the end of sort of where we go. So that started as a user project. There was a user who wrote a proposal, sent me a proposal saying, hey, can you check this draft and see if this is a viable proposal? And I said, oh yeah, that sounds really good. We should try this. We tried it, it worked. Okay, that's great. But then what came out of it was two important things. One was data processing is a big challenge. And that's why I'm currently working on exit wave reconstruction code to try and basically do that bit of the puzzle. But the second thing that we found out was Lorentz microscopy is a pain in the butt, because you have to defocus the objective lens by several hundred microns, which means your images are moving around all over the place because your lenses are just not very well aligned when you turn off the objective lens in a TEM. And so now what we are trying to do, and I don't know if this will be successful or not, is we are trying to effectively automate that process, because what ends up happening is every time you change the objective lens current, you also have to compensate for the image drift with beam shift and stage shift. And now we are trying to write code which does all three things for you. And so you don't need four hands to do Lorentz microscopy like you do right now. You can probably just hit a button and it'll work. Now, I don't know if this will work, but this is the kind of thing that ends up happening often at NSEM, where users come in through the door, we try a project, it works to some degree of success, or maybe it doesn't, but then we learn from that experience. And we think about how we can improve our capabilities at NSEM so that the next time we do a project like this, it's a lot better or a lot faster or a lot more quantitative. You can improve along many axes. So that would be my example.
Very cool. First we have Shadow. Rohan, have you met Shadow? Shadow is now the associate director, right, of AIF,
Yes.
and she has
Hi.
a question.
Yes, hi. So I'm an associate director of AIF for 2 years now. So thank you for taking your time to discuss this. So my constant, my role, and then my constant thinking is always how to make the AIF grow in terms of the technical skill or the personnel. Can you discuss a little bit about that, your career path or general the career path? For example, you have the scale for TEM from NC State, and then how do you grow yourself, and then what is the path that you generally can take at the Berkeley lab? And the second thing is your facility. Obviously, every facility has retiring instrumentation. So how do you guys adapt those things, or and then retiring it and then take an upgrade or purchasing the new I would say equipment?
Right. So I guess I'll answer the second question first because I may know a little more about that. So the way DOE funding works, I haven't been here long enough to know all the details of these things, but there is some amount of money set aside just about every year for recapitalization, and that's often major instrument upgrades, often north of a few $100,000. And also retiring old microscopes. So in the amount of time that I've been here, we have gotten a new focused ion beam, an FIB. And so there was an existing old fib. So because we were able to find a room for it, we kept the old fib, and both instruments are working in parallel right now. But often what ends up happening is you just don't find room for a new instrument. And so in that case, sometimes the instruments are moved into the basement and then they just sit there. So our basement is full of very interesting historical artifacts. Sometimes, depending on the manufacturer, if they're willing to accept a trade-in, that is also often a viable pathway, especially with Gatan. So I think recently we've had acquisitions of some high-speed cameras, and 4D STEM systems, which are all $400,000, $500,000 systems. And so these are expensive upgrades. And so often what they'll do is they'll say, hey, we'll buy your old camera back from you at a certain price. And I think that's the most efficient thing, that outcome that can happen, because otherwise often I think that retired instruments may not be utilized because we don't know how to get rid of them, basically. I think just the cost of taking a big microscope out of a lab and shipping it somewhere is so prohibitive that it's kind of hard. But I think it's very case by case, because especially with TEM labs, these instruments are so big and so expensive, they don't get changed every year, right? So they get changed every maybe 8 to 10 years or so. And so the circumstances may be very different every time each one of these upgrades happens. So I think they would definitely be open to possibilities of donating microscopes to, like, I think people have mentioned there are smaller schools near Berkeley, like CSU East Bay, and there was the thought of donating an SEM to them at some point. So there's a bunch of ways that we can get rid of old equipment that we don't want anymore, or not don't want. I would say more that we don't have the bandwidth to keep running. So the one big microscope that may end up going down that pathway sometime soon is actually a really cool microscope at NSEM. It's the Zeiss Libra. It's a energy filter TEM with a monochromator on it. It's got an omega filter. It does really beautiful FTEM and energy filter diffraction work, and monochromated imaging. But the big problem there is Zeiss no longer makes TEMs, and they don't honor service contracts for this microscope. So that means the person who handles that microscope, who was Karen Bustillo, who I mentioned before, has just a lot of extra work. And so, it'll be interesting to see what they do with that microscope, because it's a very unique TEM. It does have niche capabilities that many of our other microscopes don't, but it's a trade-off between how do you keep that in-house. And regarding my career pathway, this is kind of a bad answer, but in my experience, all academic positions are so super specialized, and the frequency of vacancies is so much. It's so infrequent compared to like if you were working as a somebody in the real world, like doing a real job that's not writing papers, and so I feel like a lot of it is luck and timing dependent. I think, as a student or a postdoc, the best thing you can do is focus on your research and make sure that your technical abilities are as good as they can possibly be. But I find, I think maybe other people have better ways of doing this, but I think hoping that you can network your way into a better career may not work so well for academia, just because it's so hard to kind of jump around from one position to another. It's all just, hey, I was graduating this year, and these 3 universities were looking for the kind of person that or the kind of skills that I have. And so those are my 3 career options, right? And sometimes instead of 3, there'll be 30 universities looking for a person like you, and sometimes there'll be 1. And so I've seen really, really talented people not find faculty positions for years and years and years. And it's not because they're not smart or talented. It's just timing, I think. And then there are other people who sometimes do better. So that's a bad answer, but I think a lot of it is luck dependent, in my opinion.
I have kind of a related question, and then we'll get to Brenda's question. So speaking to the grad students like me, is there anything you wish, like where you are now, is there anything you wish you would have known, be it technical or like life skill, that you wish you would have known when you were a grad student that you've learned in the process?
Yes, life skill for sure. So I have a mentor at Berkeley Lab whose name is Shaul Aloni, who is a well-established scientist who's worked here for like 15, 20, maybe 25 years. I don't know. He's also one of the coolest people you ever want to work with. And he once told me something very interesting, which is you should fail fast and fail often. And I think failure is part of doing research. Every time you go to the lab, you're more likely to fail than succeed. But it's also human nature to get discouraged by failures. And you have to retrain yourself to not be discouraged by failures. And I think that took me a while to learn. My first few years of grad school, I was every time something worked, I was way overexcited and thought I had invented something incredible. Nobody cared about it, and every time stuff didn't work, I would be just too depressed. And so I think that emotional aspect of science is something people should think about a little. I think that's the most important thing.
That's a really good answer, because I'm absolutely guilty of that all the time. Get way too emotionally attached to my research.
Yeah. And sometimes stuff doesn't work, and I think it's not your fault. My PhD advisor, who is like one of the most amazing people I have ever known, always told me these fun stories. So he was a student in the lab of Millie Dresselhaus, who was a famous carbon nanotube professor at MIT who passed away like a year and a half, 2 years ago, something like that. And so she had a string of students. She had prolific through the 90s. And if you're interested in reading more about Millie Dresselhaus, she's just an incredible human being. What an inspiring career. But she used to get these PhD students who would walk in through the door in year one, and in 3 years down the line, they had published like 5 papers. Each paper has 500 citations, and they go out the door within 3 years, and they have a faculty job waiting for them somewhere. And this just kept happening. There's like 7 or 8 of her students went down that kind of trajectory, and it's because they all worked very hard, but there was also an element of luck that she was working on the right thing at the right time, which was carbon nanotube optical spectroscopy, which was just blowing up. And she had already done a lot of the theoretical work to understand these spectra over the last decade. And so she was just best positioned to take advantage of the fact that now there was a group in Rice led by a guy called Richard Smalley who could make these structures. And so literally they had been working on a hypothetical structure for 20 years, and now somebody can suddenly make it, and they get samples from somebody, they look at a spectrum, they analyze the spectrum, and they have an excellent paper. But my PhD advisor took 7 years to graduate. And that's because he was working on thermoelectrics, and they were trying to find a better thermoelectric than bismuth telluride. And it's not that my PhD advisor was any less talented than any of his lab mates. It's just that ended up being a problem that still hasn't been solved satisfactorily. So it's been 25 years since then. So, I think, again, sometimes it's a bit of luck, but don't be too hard on yourself, but also make sure that you are pushing yourself to the best of your abilities.
Great. Okay, Brenda, you had a question? Oh, I think you're muted.
As I listen to you talk, of course, I have more and more questions, but I'd like to lead with, it's wonderful to see you. I had the absolute honor of being able to work with Rohan and annoy him during his time at NC State. He was always the most accommodating human being you could ever imagine. And to listen to you now, it's clear that you continue to acquire wisdom at a rate which is way beyond your years. So you're an amazing human being yourself. So a couple questions. One, listening to you describe how you're creating a solution for the annoying parts of Lorentz microscopy. You can look at a number of cases where, the old techniques of EBSD became viable techniques when technology improved to handle some of the annoying parts of it. A-star is another. And so at working at NSEM, are you or your partners, do you envision the developments you're taking with the Lorentz microscopy being able to be incorporated into some sort of commercial platform to make it more accessible to more users? Second question is, I'm really curious and really interested to hear how the pandemic has affected your region and the facility and your work.
Cool. Okay, those are excellent questions, and thank you for the really kind words, Brenda. But first question about technological development. So yes, a lot of the techniques and instruments developed at NSEM end up being things that become mainstream over time. I was just telling Aubrey a few minutes ago that I have a colleague who tells me that he was there the day that Andrei Krivenek was first able to make his spectrometer work and looked at an EL spectrum on a home-built microscope in the building next to mine and was super excited about it. And of course, now everybody has, not everybody, but a lot of people have EL spectrometers. And that's kind of one really good example of commercialization of things developed at NSEM. the other thing is the TEM project was basically the birthplace of image correction, which is another thing that's very, very mainstream now. Probe correction, image correction, both of them. The third example, historically, I just thought of something and now it's escaping me. Oh, shoot. So there have been cases that are like tremendous successes. Oh yes, the third example was there's a guy at our synchrotron lab called Peter Dennis, who is a very well-established scientist who was one of the instrumental people in developing direct electron detectors, which has basically enabled a lot of microscopy over the last 10 years. I would argue that cryo-electron microscopy has been enabled only because of that development. And so that's another example. Where things go based on what we are working on, I think most of the people at Berkeley Lab are not in it to make a lot of money. But I feel like oftentimes people would leave the technology at an open source kind of phase, and they would say, oh, this thing works. I have written the code. It's on GitHub. You can look at it and you can design your own setup accordingly. But those are always buggy. So one case is like the MPAD, for example, is at NC State, and some of you may have used it, and it's basically a few grad students and postdocs, research project to develop that. And I think I don't know if any of the things we are working on will reach that level of success. Hopefully, but we can only try. But there have been several examples of very successful technology transfers from NSEM in the past. And regarding the pandemic, it's been very interesting. So the governor of California was reasonably proactive in calling a lockdown across the state. But I think actually even before him, all the Bay Area mayors, so of the different counties in the Bay Area, called a shelter in place. And I think this was somewhere in March. So that day was kind of crazy because, of course, everybody knew, oh, coronavirus everywhere, be careful, stuff like that. But we didn't know that the lab was going to be shut down. And then at 11:30, like our facility director came charging into the lab, like driving his car full speed, and he was busy somewhere else, and he's like, shut it down. It was like a movie sequence. And so we had to do a very quick lab shutdown. And then we had to all go home because the San Francisco mayor declared a shelter in place on that day. And so then, of course, there was like, oh, what do we do now? So firstly, everybody started working on Zoom. And then there have been, so lab management also does a phenomenal job of thinking through a lot of these issues. And so they've been working on a plan for the last month or so. And now they are going to start on June 1st, they're going to start reopening labs, but it'll be in a very, very controlled fashion. It's going to be a multi-phase approach. So phase one will just be some fraction of the NSEM or Berkeley Lab staff going back onto campus. And then phase two will be maybe increasing that number by a certain amount. And then eventually we'll start taking in external users again. I know a lot of the advanced light sources and advanced photon sources and synchrotrons have just said no users till the end of the year. But everything's going to be done with the point of view of we are going to very carefully regulate how many people are there in a building at any given point in time. They'll have spray bottles and of disinfectant everywhere. So no matter what you're doing, you're going to disinfect the surfaces you're working on. Everybody is going to be required to wear some kind of face mask. So all those kinds of precautions, and there's actually a long email that's been sent across by the lab management saying these are the things you must do. The other really good thing they've done is before we start work on June 1st, we'll all have to take a COVID training. which is an online training. It's kind of like we have something called the Gert, which is a radiation safety training program that we must do once a year, or we lose our badge access to all the rooms. And I think it's kind of going to be similar, where everybody, it doesn't matter if you're a user or a scientist, or even like say a person from FEI or JEOL who's coming to fix a microscope. Even they need to go through that online training before they can come on and do subcontractor work. And so I think that will be really useful in sort of getting everybody on the same page, so that it's not like, there's 9 of us who are taking all these kinds of precautions, and then there's this one guy who's, going around licking the doorknobs or something like that. And then of course, they're going to monitor it closely. So our facility director has sort of said, if somehow like a few cases start showing up in the lab, I think they're going to very quickly shut things down again. So it's definitely impacted the way we work, just like everybody else. The only good thing is that all of our management is full of scientists, and I trust their thinking more than a lot of other institutions have managers who are, have economic incentives to look at. And so I think the decision making goes a little weird. And there was one other thing that I guess I'm forgetting. But yeah, I think that's where we are right now. Oh yeah, the other thing that's interesting is, so in the Bay Area last year, we had a bunch of forest fires, which is very unfortunate, but they cut off power to a lot of the Bay Area for almost a month. And for me personally, I feel like that experience kind of prepared me for working from home, because firstly, we got really, really good at safely switching off all of our microscopes or putting them in a standby mode. So literally that day when our boss came in and said, shut everything down, within 45 minutes, 3 people at NSEM shut down like 12 microscopes, and it's like, okay, done. We know exactly what to do. And I think that's kind of been almost a dry run for this whole experience. So we've sort of done a reasonable job so far of handling it.
Thank you.
Yeah, thanks. So we're nearing the hour mark here. So I have 2 more short questions, and then I'll stop the recording, and then we can hang out and Rohan can answer our questions as long as he's willing to. So 2 kind of quick ones. First, what's your favorite TEM technique? Like your favorite kind of experiment?
I like all in situ experiments because they are hard and you are more likely to fail. And I don't know, I'm always attracted towards hard problems. It's sometimes not a good thing, because from time to time, it's just nice to take an easy win. But I really enjoy in situ microscopy, and I think Lorentz has been a lot of fun. But honestly, I'm going to give you a really boring answer. Electron diffraction. I still have to reread, basic like Williams and Carter once
like
every 6 months. And
spot patterns.
yeah, so I think I still have a lot to learn in electron diffraction. And like, I find it funny that people are doing, for example, 4D STEM, where it's 4D STEM is like saying, I don't want to think about what I'm going to do beforehand. So I'm just going to collect all the data and then think about it afterwards. There's more than that. You can do a lot more than that. I should not be that.
It's like going to the smorgasbord. It's all right there.
Yeah, but yeah, exactly. I think a lot of the old school TEM, the problem is with the educational pipeline, because as we can do probe corrected STEM, or as we can do 4D STEM, people forget how to do like old school electron diffraction. And that would be my answer, is electron diffraction.
You just made a lot of enemies of 4D STEM people.
So I say this as somebody who also does 4D STEM and is currently analyzing 4D STEM data. I mean, I get why it's awesome, and it is awesome. It's 100% awesome. But I think it should not come at the cost of knowing how to do parallel beam electron diffraction first.
Good answer. Okay, so last quick question. What are you doing to stay sane while social distancing?
I have a lot of weird projects. You can see my 3D printer back there. I can keep myself busy. I've been making these silly things. These are Arduino thingies. And so I found these $4 particulate matter sensors online, which are amazing because so the lab also does a lot of outreach work with local school districts, and at some point, and I like that kind of stuff. So at some point, I proposed that we build air quality sensors and teach students how to program them with a small Arduino kit, and then how to plot the data out, and then maybe calculate a simple moving average of the data so that we tell them about noise versus signal and things like that. And so this is another reason why Berkeley Lab is such an interesting place to work, because you can find people doing all kinds of crazy stuff. And so that we had a K through 12 coordinator who sent out an email and said, hey, is anybody interested in talking to local middle schools and high schools and coming up with science projects for them? And I said, sure, how about we do this? So literally, I have 3 more meetings today, and one of them is with this K through 12 coordinator who wants me to teach the middle school teachers how to program this thing, and they've already spoken to the company that makes that sensor, and they'll get them for free because it's really cheap. And so that's what I've been doing. I tore apart a bicycle and put an electric motor on it.
So you're still an electrical engineer at heart.
I definitely am an engineer. So I've been doing a lot of reading. Which is nice, because I think I used to really like reading books, and then the last 10 years of my life are just like, you read papers and that's it. Like, you don't want to see anything else, right? So I think it's been kind of nice that I've been able to read some lots of nonfiction stuff. Yeah,
right.
That kind of stuff.
Can we get a cameo from Anyang?
You want to say
hi?
Okay, so he's busy. This is my very messy apartment, but you see him at the window. And so we just sort of moved into this apartment a few weeks ago, and that window faces the courtyard of the building. And so literally all of my neighbors walk past, and I catch people like petting the cat through the window mesh. And he's extremely popular in the building because he will let anybody pet him. So, but right now he's having his afternoon snooze. He had a hard night of work, I think. I don't know. Hard night of snoozing followed by afternoon snooze.
Anyang is quite famous in his own right.
Yeah.
Well, thank you guys so much. Yeah. Thank you guys. Thank you for all your questions. Rohan, thank you so much for being willing to do this.
For sure.
Feel free. I'm gonna cut us here, but feel free to hang out, and Rohan will answer.
I'd just like to say one quick thing before we go is, all the students and postdocs who are on this chat, like, I think you should at least be aware of the NSRC research programs and what an amazing resource they are. It's really hard to find access to instruments like this through any other mechanism. And of course, NSEM has a set of technical skills that are always evolving. I was just telling Aubrey the latest and most amazing things we are doing is we have an insane 100,000 frames per second 4D STEM detector that's just been installed. And we have on our team one, we can now do energy filtered 4D STEM, which is really cool for thick samples. But these are typically things that you won't find in most other labs. And I think if you have a research project that needs these kinds of capabilities, you should not be shy. Reach out to scientific staff at NSEM or at Berkeley Lab. Everybody will help you. And yeah, I think that's really important. And if you have questions about what we are doing at NSEM, feel free to email me. My email address, I don't know, I think Aubrey, I can hand it out to everybody. I'm happy to answer questions. But I think for us, our bread and butter is actually users coming in through the door with interesting research problems, and we help them make it happen. And so, you have to give us that opportunity first.
Nice closing plug for your job there.
Mm-hmm.
All right. Well, thank you all so much. That is all for this week's user meeting, and be looking out for our next one.